A pleasant afternoon to all. It is my great pleasure to welcome you to the opening of the OMICSena lecture series. Some of you might have attended first OMICSena last March. That was an information field premiere of the OMICSena platform where DOST-FNI featured the frequently asked questions on nutrigenomics. For the second time, DOST-FNI brings you OMICSena with a bolder move to bring together omics experts from way beyond the Philippine borders. The Philippines continues to build strong innovative foundations, and learning and reflecting from the global scene is a very way to go. As such, we are thrilled to bring you the 9-day OMICSena lecture series. For 9 afternoons, we are expecting to learn the best of omics technologies and how these emerging platforms can enhance nutrition and food research that will eventually be translated for societal benefits of our country. Beyond basics, hopes are high that we can also build networks and collaborative paths, all towards building an omic-driven, innovative Philippines. William Wordsworth said that life is divided into 3 terms: that which was, which is, and which will be. So let us learn from the past to profit by the present, and from the present to live better in the future. I hope that through all the topics to be discussed from this day until October 7, we can learn how to create a better future in life and nutrition through the studies of omics sciences. To all the participants, again, my warmest welcome, and lend us your active participation because we have inspiring lecture series ahead. So thank you so much and stay tuned. Maraming salamat po at magandang araw muli.
Magandang agham po sa ating lahat. OMICSena was coined by DOST-FNRI to create a scene in a good way. It seems that it will continue to serve as a platform for the DOST-FNRI to keep us in the loop on its undertaking in genomics. With Dr. Brian Gonzalez on board, the second OMICSena for 2021 collaborates with the DOST's Balik Scientist program to drumbeat the advent of another set of omics science at the Institute, the metabolomics. This expansion in the R&D platform will guide DOST-FNRI in setting up more actionable strategies in the molecular food and nutrition arena. I am optimistic that the series will provide new learnings to bring stronger and bolder omics-based research and development initiatives, not only in DOST-FNRI, but in the entire country, as I understand that this OMICSena is well represented by genomics enthusiasts mula Luzon, Visayas, at Mindanao. As we come together for the next 9 afternoons, I hope that we will be able to lay out plans and strategies towards the creation of new intervention systems, biomarker discovery, and innovative health and nutrition platforms through the use of omics technologies. As we come together, this event sharpen our vision to acquire the lens of seeing through what our country needs to reach zero hunger and optimum health and nutrition at the molecular level. Congratulations to the organizing committee of the OMICSena lecture series and to all the staff of DOST-FNRI led by Dr. Imelda Angeles Agdepa. Magandang agham sa ating lahat.
A pleasant day to all. The Nutritional Genomics Program of the DOST-Food and Nutrition Research Institute is part of the DOST's Big 21 in 2021. This achievement highlights the growing field of nutritional genomics and the DOST-FNRI's New Gen Laboratory, now an ISO 17025 Certified Molecular Laboratory. Beyond this recognition, I am more thrilled to see what our R&D program and scientific and technological services in nutritional genomics can offer to ensure optimum nutrition for everyone. Earlier this year, the first OMICSena answered frequently asked questions on nutrigenomics. The first installment of this web-based learning platform of DOST-FNRI's genomics programs made us realize that nutritional genomics can scale up nutrition and health sciences. Studying our genes through the omics platforms can bring about enhanced nutrition recommendations and perhaps nutrition and health policies in the long run. Its eventual integration in clinical practice can boost our efforts to overcome malnutrition and help us achieve our goals of good health and well-being. As DOST continues to expand the country's omics footprint, this second offering of OMICSena with Dr. Brian Gonzalez, our balik scientist for metabolomics, is anticipated to bring new frontiers in omics-based innovations. This will fine-tune our efforts to use various molecular platforms to pacify and eventually end our long-standing battle against malnutrition and non-communicable diseases. Now, with the generous sharing of information and practices from our keynote speakers ahead of us, I invite you all to join me in getting a detailed glance at omics technologies and how they can play critical roles in nutrition and health. May this deep appreciation of omics technologies guide us in pushing for more innovations directed to our local needs. I am optimistic that this lecture series will also strengthen the competencies of our researchers in initiating omics-based research and development. One thing that the ongoing pandemic has taught us is to hold tight to what science and technology can do. Science and technology will always be an important key in solving various societal problems. Napaghahandaan man o hindi, katulad nitong pandemya, innovations and discoveries will help us thrive and become resilient. On behalf of the Department of Science and Technology, I would like to thank our speakers from different parts of the European Union and the United Kingdom for their time and expertise. I extend my appreciation especially to our DOST Balik scientist, Dr. Gerard Brian L. Gonzalez, for bringing this exciting series to light, together with the DOST Food and Nutrition Research Institute. Thank you all for being here. Let us have a learning-filled week ahead. Mabuhay!
To a place where A pleasant afternoon here in the Philippines, and a pleasant morning to our speakers and viewers from the Netherlands and United Kingdom. Welcome to the third day of the OMICSena lecture series. I am Frances Isabel Hacalan, and I will be your moderator for today's lecture. The OMICSena webinar series is part of our 75th DOST-FNRI anniversary countdown. We started our countdown this month, and we have 10 months to go before our 75th anniversary next year. As we have been in every household of every Filipino, our tagline is DOST-FNRI, kasama nyo sa bawat yugto ng buhay at kabuhayan. At this point, allow me to acknowledge the presence of our former DOST-FNRI Director, Dr. Mario Capensana. Maagham na hapon po sa inyo, sir. It's day 3 rather, of 9 of our lecture series, and we have 2 topics to learn today. Our first topic will be given by Dr. Gordana Panich. Dr. Panich completed her PhD at the Swiss Tropical and Public Health Institute in Basel, Switzerland, where she focused on preclinical and clinical drug discovery against parasitic helminths and the metabolic fingerprints they impart on their host. As a research fellow at Southampton University, she is applying both untargeted nuclear magnetic resonance and targeted mass spectrometry to investigate the metabolic relationship between undernutrition, infections, and the structural and microbial enteric environment, and how these interplays influence growth and health outcomes. To discuss about metabolic profiling for biomarkers of enteric infections and malnutrition, ladies and gentlemen, please welcome on the virtual platform, Dr. Gordana Panich. Over to you, Dr. Panich.
Thank you very much, Frances. I'm just wonderful. Thank you. I'm just going to start sharing my screen. So actually, before I start, I just really wanted to thank everybody for attending today. It's very rare nowadays to have conferences live, but on the upside, there are more people that can join. So I'm so glad that so many people have joined in today. Before I get started, I just want to encourage you, if you feel like, please feel free to have your camera on, just have your microphone off. If you prefer to see faces other than myself. and I definitely enjoy seeing your faces. Yeah, I just want to thank Brian for organizing this wonderful conference. So he just needed to say the words capacity building, and I was there. Okay, so one second.
We also would like to thank you, Gordana, for accepting our invitation to be one of our lecturers.
Thank you, Frances. Yeah, like I said, it was really he just needed to say metabolomics and capacity building, and that was that was the key entry. It was great to chat with Brian as well. So,
Okay.
Basically, what I'm going to do today is talk to you about some of the work that I've been doing over the past while. And what it essentially will be is kind of like a sampling of some of the things you can do with actually both untargeted and targeted metabolomics. So basically, I will take you through 2 studies that I've been conducting over the last while and what they've been revealing. So just having there we go. Yeah, and in the spirit of what Brian did at the beginning of the conference, I like that he introduced not only himself, but where he is from and where that is in the globe. And so I know a lot of people know where the UK is, but maybe some people are a bit less familiar with Southampton. It's just a little bit south of London. And it's a great place to be. It's a seaside city, it's a port city. And both myself and my advisor, Dr. Jonathan Swan, who will be speaking later on in the conference, we were both actually working at Imperial College London before this in London. And John relocated to Southampton because there was a great opportunity there, as he had a chance to integrate his metabolomics work within the clinical setting there. So yeah, when he offered for me to join, I gladly said yes. So you're going to be, you've already heard a really, really nice overview from Brian on Monday, but just in case some people couldn't join, just a quick, very, very quick overview of metabolomics. So metabolomics is kind of a logical extension of the various other omics that are used to study the organism. And just reiterating a little bit what Brian said, genomics, it tells us about what's in our DNA, which is what can happen. Transcriptomics is a bit more about what may be happening. And when we measure the whole protein content, we see that what's making it happen. And finally, metabolomics or metabolomics is basically what has happened. For a lot of people, that's pretty clear, but just a quick reminder of what metabolomics measures. It measures metabolites, which are any organic molecule that are less than 1,500 Daltons. So a more simple explanation is basically all the organic stuff that makes your metabolism happen, the chemistry of your body. Anything like glucose can be a metabolite, or uracil, etc. And the metabolites are kind of a byproduct of all the things that are coming in from the environment. But as well, they're used as biomarkers of disease or treatment, and they can also elucidate mechanisms for disease or treatment. You can study metabolomics in two different ways. We can do untargeted metabolic profiling, which basically means that you're not looking for any specific thing. So this is you're just profiling everything and seeing how the whole profile changes according to, for example, presence or absence of disease. And the tools we have for that are the various mass spectrometry in tandem tools, as well as nuclear magnetic resonance spectroscopy. And there, basically, because we profile everything, we also have to employ quite a lot of statistical tools to normalize all the signals. And then we rather use multivariate statistics to sort of look at pathways. And as a result, a lot of the times this is more hypothesis driven. On the other hand, you can generate a targeted metabolic profile, and that's where basically you know exactly what it is you're looking for. Not only do you know what it is, do you want to detect it, you want to quantify it. And with that, we tend to then use more classic univariate statistics, t-tests, ANOVAs, etc., in order to quantify the response. So what I'm going to talk about is, I'm just going to see if I can get this out of the way. Oh, no, that's all right. So in this particular talk, by the way, everybody can see my slides, yes?
Yes.
Yes.
Yeah, okay, great. Thank you. So this is a study that I started a long time ago during my PhD. And I was looking into the metabolic fingerprints of schistosomiasis, which is a disease that some of you may know about, but some not. So I'll quickly overview it. This was actually my first introduction to metabolomics, but it was actually also my first introduction to my current advisor, Dr. Jonathan Swan. This was actually a really sort of serendipitous jumping off point. So in case you need a little bit of an overview, oh, so sorry, we just tested the slides a minute ago and that seemed to work fine. Okay, there we go. So schistosomiasis is actually a very highly prevalent disease worldwide. So about 142 million people that are affected. And because it's more so a chronic disease, we don't actually measure its impact in terms of number of deaths. It's more like the years of life lived with disability, which is total of about 1.1 million years of life lived with disability. It's caused by this parasite, as you may have seen, a blood fluke or a worm of the Schistosoma species. And they're generally spread out in the tropics and subtropics, and usually in rural areas of poor sanitation. And as you can see, there's different species according to the geography. So in the Philippines, what's mostly prevalent is Schistosomia japonicum. And as you can see, it is spread in many areas. The national prevalence is 4.68%, which may not sound like a lot, but I would argue that if that was a percentage of COVID, we would all be a little bit worried. But to give the national prevalence is very misleading because schistosomiasis is actually a very focal disease. So you have certain areas in barangays that have a higher prevalence than 5%, sometimes even up to 60%. And on top of that, what's special about areas that have Schistosomia japonicum as their infective agent is that they can actually also infect rats or dogs or the water buffalo. And actually these animals act as an important reservoir. So as a result, it is estimated that the prevalence of schistosomiasis is actually highly underreported and underestimated. And this is sort of the more projected risk as of 2014. So I always love giving an overview of this slide because my initial jump into this study was actually the parasites themselves. They're quite freaky. They transport themselves over and over again. So the way you become infected is basically if a human comes into contact with fresh water that's contaminated with Cercaria, then these Cercaria basically, it's not just by chance, they actually go and by chemotaxis sense out a human and penetrate through the skin where the tail breaks off, and then you have these microscopic schistosomia that wander through the circulatory system, travel through the lungs and end up in the liver. And this is where they actually sexualize, so they become male or female. And they mature and then they pair up. And finally, they migrate to their final site. And if it's Schistosomia japonicum or Mansoni, then it's going to be in the veins of the mesentery. They're going to live there as a couple for many, many years. Or if it's urinary schistosomiasis, then it's going to be in the areas of the bladder or the genitals. And so, like I said, with in Asia, it's a little bit special because humans can contribute to this pool of Cercaria, but so can other animals like the water buffalo. So it's not the worms themselves that give the disease. At least that's what is thought of currently. It's more actually the eggs that they lay, because the eggs that they lay elicit a very strong immune response. Initially, a very strong acute response, and with early onset of neutrophils and basophils, but also actually quite a lot of T cells and B cells. And then afterwards, what you get is this kind of hard fibrosing with still a lot of immune cells trying to resolve both the presence of the egg and the resulting fibrosis. And that would be, you know, manageable if it was just 1 or 2 eggs, but actually a lot of species can lay anywhere from 10 to hundreds of eggs per day. And half of them are going to get shed into the water, but the other half are going to get stuck in the intestinal wall, in the liver, in the bladder area. And so slowly the organs start to fibrose over many, many years. And often it goes undetected. So yeah, here you see some of the pathology. This is the normal liver and a fibrosed liver. And here below are ultrasounds of the bladder where you actually start to see polyps. So the manifestation of the disease, usually the acute phase is in most species, if you're living in an endemic area, you're not likely to experience acute phase just because of all the repeated exposure. Actually, with Schistosomia japonicum, you experience it quite a bit more, but it looks a lot like fever, very, very, very strong fever. And then you get over it. But then the chronic phase, as a result of the many eggs that get laid over many years, results in all kinds of complications, including eventually for urinary schistosomiasis, bladder cancer, or infertility. And this is indeed then categorized as one of the diseases that are quote unquote the diseases of poverty, because if you have this, you're likely living in a rural area that it's not well serviced, that doesn't have ideal hygienic infrastructure, and potentially you're malnourished, so you're susceptible to these infections. But then actually acquiring these infections also perpetuates further malnutrition and growth stunting and cognitive inhibition. The control strategy is pretty comprehensive. So of course, hygiene measures to break the cycle. Here you see a man spraying molluscicides to get rid of the vector. And of course, one thing that actually the Philippines has done quite early is to integrate animal health into the control program. And this is done under the One Health lens. However, I get asked a lot, why can't we just, implement proper hygiene and that'll be the end of it? And it's not that easy, because wash measures, as anyone who works in this field knows, are notoriously difficult to not only implement, but to maintain. And not everybody's on board with the measures. Vector control can help, but snails are actually pretty tricky. They will hide and they will come back when it's the right time. And yeah, One Health is very important, but it can be difficult to convince all the farmers to get their animals treated. So actually a really, really, really important aspect of controlling schistosomiasis is administrating the current only effective treatment called Preziquantal. Preziquantal is effective against all the species. It's not 100% effective, but it does a pretty good job at reducing the worm burden. And actually most control programs, they don't wait to diagnose someone, because it's very difficult to get to rural areas. And that would be actually very inefficient. So either once or twice a year, they go into villages and the population receives a prophylactic Preziquantal treatment. And in that way, they hope to catch everybody. And here I'm showing a picture of a child, because actually for most cases, schistosomiasis affects mostly children. This is less true in the Philippines, just because they're actually more so farmers and people working in water. But actually children are still very highly affected. And so children are kind of like the target population. A lot of the administration is done in schools. So lots of nations have set targets to eliminate schistosomiasis. And in the Philippines, the target has been set by 2025. And the reason they specifically put this in place is because a lot of areas appear to have low prevalence, and we actually have a lot of the tools. But as I was saying before, because of underreporting, it's although a lot has been done to reduce schistosomiasis in the region, it's not on track for elimination at the moment, and that's not unique to the Philippines. That's actually the case for a lot of nations. So one then important aspect is really making sure that most infants can take the medication that they need. As you can see, the actual tablets are actually quite huge. And one thing some of you may know if you've ever had to take it is Preziquantal is incredibly bitter. It's actually the standard used in pharmacy to measure bitterness against. So it's actually very difficult for children to take it well, even when their mothers, do their thing and and force them. It's actually, quite difficult for them to take it. And so a while ago, the Pediatric Preziquantal Consortium was formed in order to make a much more, not only a more palatable formulation, so a smaller one and less bitter, but actually also to figure out what exactly is the right dose for children, because to date, it's just been the correct dose was determined for adults, and then for children it was just back calculated. But children are not just small adults; they're very different organisms, and they process this very differently. And so basically, this is Preziquantal. This is what it looks like. The other element of it is that the tablet is made up of 2 enantiomers of the drug. So it's the same molecule, but it's a mirror image of each other. And the thing is, actually, our Preziquantal is the enantiomer that carries the efficacy. And S Preziquantal is thought to be less effective, even though it's much better absorbed. And it's also the thing that's thought to carry the bitter taste. So they basically wanted to make an enantio pure formulation where they just make our Preziquantal. But to do that, you need to figure out what is the right dosing and formulation. So basically, a few years ago, the team that I was working with then during my PhD at the Swiss Tropical Institute initiated a clinical trial in Ivory Coast where we have one of our partner institutes to essentially, determine just that. What is the effective dose? Is there an effective dose for young children? And can we actually realistically make an effective formulation with just our Preziquantal? And so, this was my former supervisor, Jennifer Kaiser, together with the lead of the team in Ivory Coast, Dr. Jean Koulibaly, 2 amazing people who never quit, never ever quit, especially Jean. He's just got such a good spirit about him. So this is the team that they led this particular trial, and this is just a picture of just some of the team that it took to get this underway. As I go along, I'm going to try and what I've done is just try and put in some pictures of people that are involved. I know it's typical to put in acknowledgements in the end, but I just feel like it's kind of important that we know that people actually do this work, and it's really hard work. And sometimes I think when you just read names at the end, you don't really make the association. So this is kind of why I'm doing that. So off we went, and basically I got to join the team because we also had the question of how does actually schistosomiasis affect the organism, basically the human host, metabolically speaking? Does it actually leave any metabolic fingerprints? And do those go away after treatment? So Just as a quick overview of the study design, this was a group of school-age children and preschool-age children that were screened and randomized to 4 different treatment arms. Basically, this was the doses that were sort of deemed less effective to maybe maximally effective as safe. And there was then also a placebo to compare it to. And usually we actually don't anymore compare drugs to a placebo. But in this case, we needed to do it for the dosing purposes. And the reason we could do it is because essentially 3 weeks after the whole follow-up, everybody gets Preziquantal anyway, is treated. So the part where I came along was I collected urine specifically prior to treatment, 24-hour post-treatment, and 3 weeks when we were doing the follow-up and evaluation. And this is just a little bit of some of the sample collection. So yeah, the urine was collected, and then basically after this preparation, I needed to run it through this wonderful little nuclear magnetic resonance machine with an automatic sampler. And basically, we just did a one-dimensional scan on it. These are very technical terms. I'll explain something in a minute. Then afterwards, what you get is these spectra, and here they look nice and neat, but initially, actually, they're kind of all out of order. You have to do, and they're all out of order up and down, so you have to do a bit of baseline correction, you have to adjust their phasing, you have to reference it to an internal standard that we put in and normalize it. And finally, you can do the data analysis on it. And like I said, it's usually because you're collecting everything, then you can't just do univariate statistics. You have to employ type of statistics where you take everything into account, but in the end, you want to filter out all the noise and just look at the features that matter. In this talk, I'm not going to go too much into detail about how we do this because in October 5th, my advisor, Dr. Jonathan Swan, will be going exactly into how to do this, and he will give it in very nice detail. So I would highly encourage you to attend that. John, if you're there, sorry, I know this is not your title slide, but this is it just matches mine. All right. So this was actually done like over 2 studies. We did in one study was just looking at intestinal schistosomiasis, and by intestinal I just mean people infected with Schistosoma monsooni, because that is the only form of intestinal schistosomiasis in Ivory Coast. And basically we simply looked at what was up or down regulated as a result of being infected or not infected. So we had a group of matching negative controls. And in here, what we can see is that you don't, for intestinal schistosomiasis, you don't get a whole lot of metabolites that are indicative of infection, but you get a few. These here are part of the TCA cycle, as well as Fumarate microbial. But mainly what you see is essentially a bit of a depletion of certain microbial metabolites. Creatinine is not actually a microbial metabolite. It's related to muscle mass. And here mannitol, which was initially we thought that's a little bit strange, but actually turns out that the Ivorian diet is pretty high in mannitol, and that's a pretty good indicator of a leaky gut. So a lower amount of mannitol in the urine as a result of infections could indicate that we don't have proper absorption. And then, of course, a few more that were sort of unknown at the time. And that's the infection column. We were just kind of interested in how it relates to age, so I wouldn't pay as much attention to that. But next, we basically wanted to see, do we get clearance of, do we have opposite effect on these metabolites when we dose with Preziquantal? And does it go in exactly the opposite direction? And one thing we were kind of perplexed to find is that basically the metabolic profile after treatment, both 24 hours and 3 weeks follow-up, didn't really have much to do with any of the metabolites found to be associated with infection, except for maybe a bit of trimethylamine. But they weren't exactly complementary, and we actually found that that's a bit disappointing, because you'd like to see, you know, as a scientist, look, I found this effect, and when I take it away, then you see the opposite effect. But the thing about schistosomiasis is once you take away the pathogen, the actual damage that the pathogen has caused does not go away immediately. It doesn't even go away in a few months. You know, it can take a lot of time to resolve. So actually the signatures that we're seeing here, we postulated don't really have much to do with infection clearance. It's actually more how the host is reacting to the tablet itself. And this will become relevant in a little bit. And then in a separate trial, we did the same for urinary schistosomiasis. And, you know, the signatures for urinary schistosomiasis are not at all the same as for intestinal. There are many other signatures going on. One thing that we can sort of say is in common is that you have, again, a little bit of this effect of depletion of microbial metabolites, but it's generally speaking a whole other ballgame. And one thing that actually we also see again here is that when the children are given Preziquantal, this is just the 24-hour, but also that follow-up. The metabolic profile there doesn't really have anything to do with the infection. So again, we're postulating that this is rather just metabolic signatures of the treatment itself. So we wanted to, you get this metabolic signature and you think, okay, so what can I do with that? Can I associate it with anything? In the case of urinary schistosomiasis, we were lucky because for other reasons, we had my colleague Beatrice Barda seen here, who was looking at basically evaluating if a portable ultrasound device can be used to assess morbidity in children in urinary schistosomiasis. And so she actually, she examined all the children before and after treatment, and was able to sort of generate a table of all kinds of parameters that would indicate morbidity. So things like presence of polyps, the volume of the bladder, before and after, alteration to the bladder wall, and basically the level of alteration. Now these numbers here are my apologies, they're a little bit confounding, but basically it's how we measure whether the models are significant or not. And I can tell you here, you can see a p-value. Most of the time, we weren't able to associate any metabolites with morbidity, except if we have sort of an aggregate score of alteration to the bladder wall. And so there we found that actually significant associations that here I'm just showing you a different way of demonstrating this I guess the great thing is here, everything that points up is associated with a higher score of alteration grade, and everything that points down is associated with a lower score. And basically we found that the changes here were actually pretty consistent with the changes that we were seeing as a result of infection. So that was exciting. We didn't have these kinds of measures in place for the intestinal schistosomiasis. However, I'm really glad that at the moment I'm working with two lovely ladies from the University of Manchester who have a wonderful animal model, and in which they can, they've been collecting samples from all different compartments at all different time points, and in different kinds of infection modes, so we can sort of suss out the association between the metabolites and the different compartments. So I was telling you a little bit before that it was confounding for us. It was very confusing for us to see that the metabolic profile of the children was pretty different after Preziquantal treatment, and just not at all really correlating with the infection profile. Yep, sorry. And so we actually, afterwards, we actually then had a pretty strong idea. One of the things we really need to know is basically, is there a good dose of, proper dose of Preziquantal that will give us a good cure? And is it properly getting into the bloodstream and where it needs to go? And to assess that, actually, what you need to be doing is pharmacokinetics. So basically, after the children receive their treatment, their blood is sampled, and we had a very non-invasive dried blood spot sampling. And you see how the concentration of the blood, concentration of the drug varies over time in the plasma. So how long it stays there, and how long it takes to get eliminated, and how high the concentration goes. And basically to see if any of these parameters actually correlate with cure. And so this work was done by my colleague Jana Kovac, and she was able to generate these pharmacokinetic profiles. And my thought was like, okay, we learned a little bit of something about how much concentration you need to reach of the right enantiomer of the drug in order to get a curative dose. But the thing is, like, you see these zigzaggy patterns here. That's actually something we constantly see. It's actually pretty difficult to measure Preziquantal smoothly in the plasma. It's completely erratic in all the children. And we have the suspicion that it has a lot to do with the children's own status. So because we already had the data, I decided, okay, we'll combine the metabolic profile together with the pharmacokinetic profile. And that's when you do this together, that's something called pharmacometabolomics. And you can try to gain insight about what are the metabolic interactions with the drug, and also if we have predictive biomarkers. And just for the interest of time, and I know people here don't really want to get into pharmacy, but one of the things we were able to do is basically demonstrate which metabolites in the urine were prior to treatment were predictive of higher Preziquantal levels in the plasma. So here, basically we see that histamine and methylhistidine is quite well associated with more of the drug, as well as a few other metabolites. And we then pulled this out and just wanted to see, like, if there's a consistent, something consistently between all the different enantiomers. And basically what we saw was that in cured children, you had higher levels of alanine and creatinine and acetone as in uncured children. So this, together with some of the other metabolites that we see here, suggests that there is potentially a role, some of these metabolites are microbial metabolites. So there's potential role for the microbiome here. And so the work that we're currently doing is my colleague Pierre Schneeberger, whose pictures I forgot to put up. I'm sorry, Pierre. He has done on this very same samples, he's generated a microbial profile. Together, we're putting together our findings, and we hope to have something out for that soon. Okay, now that was a little bit of taste of what we were doing with untargeted metabolomics. I'll give you another study here with targeted, and basically, I think here for a lot of you here, I don't need to explain this too much. You already know that lots of places in the world, especially also in the Philippines, children experience malnutrition, and it's not just about whether you have enough food. It's this whole environment where the exposure to infections makes you less well to absorb nutrients, which affects the microbiome and immunity. And altogether, it affects basically the growth and developmental outcomes of children. And this kind of then goes on and on between generations and is to do something. And a lot of the world nutritional interventions have helped in bringing down the rate of malnutrition, but they haven't been enough. There's an estimate that if we just scaled everything up, all the nutritions we have so far, and had 90% coverage, we would only manage to reduce the remaining malnutrition numbers by 20%. So clearly, it's not just about caloric intake, and it's not just about nutrient intake. So the team of Andy Prendergast and Robert Nzini in the Zwitfame Institute in Zimbabwe started a trial a while ago, the SHINE trial, in which basically they had enrolled mothers and checked in on them 20 weeks and followed their infants up to 18 months and gave them an intervention. So the control being like a basic standard of care where they promote exclusive breastfeeding, provide some micronutrients, and offer lots of village health worker support. Then another group got a wash intervention. I think a lot of you know what that looks like, but just as a revamp, it's, all the community gets access to latrines, and not just any stinky latrine, good, clean, ventilated latrines, lots of sanitation stations, just like you saw in the previous picture, access to clean water, to water cleaning agents, soap, and then of course promotion of hygiene with the health workers coming in to check regularly. Another group got a nutritional supplement, NutriButter. I think a lot of you know about that one, which contains basically a lot of micro and macronutrients. So they received this at 20 grams per day, and they would also advise how to best use their local food. And finally, another group got both interventions, and everyone got the same standard of care. And this was randomized by region, because as you can imagine, randomizing it in a same village is not so easy. So where do we fit in all this? Well, basically, we were asked, or we had suggested that that we measure the metabolites that are part of this pathway. And I know that can look complicated, but basically this is the tryptophan degradation pathway. That is one of the main pathways that generates NAD+. And NAD+ is a very, very important molecule in the body for energy, for all kinds of biochemical reactions. You need it as much as you need ATP. You can also get it from the diet. So a diet that's rich in nicotinic acid. And on top of that, there's this salvage pathway where when the NAD+ gets broken down, you can actually it can actually be recycled with the addition, with the help of dietary supplementation back into NAD. So why is this pathway important? Well, aside from making sure that individuals have enough tryptophan in their diet, and tryptophan is an essential amino acid that's actually more frequent in more protein holding foods like like meats and dairy and so forth. Aside from that, this particular tryptophan pathway can go awry when you have local inflammation, because what that does is activate, instead of the usual TDO, it activates this IDO enzyme, which quickly causes tryptophan to degrade. And then a lot of the tryptophan doesn't actually go down this essential pathway. It's actually getting diverted into all these different side molecules that help try to deal with whatever is causing the inflammation. So yeah, that's just a little immune primer for you. And what John and Jordy did in a few studies ago in a Brazilian cohort was establish that essentially a lot of the inflammation markers that he collected were associated with basically an increase in this KT ratio, which is indicative of this inflammation pathway. And that also that the KT ratio was higher in stunted children. So basically in the SHINE trial, one of our jobs was to measure the metabolites of this pathway. And so this is just it, more right side up. So everything in green is what we were able to measure. So that I think is pretty straightforward. But on top of that, we actually also measured quite a lot of these side pathways. So basically where the kynurenine can get diverted away from the NAD+. And these molecules in themselves, as you can see, have important functions. So it was worth measuring them. And just as a form of control, we included inflammation marker just to neopterin, just to see if general if inflammation is happening. Citrulline, which is a gut health marker, it's indicative both of enterocyte mass and function. And finally, indole-3AA, which is basically generated as a result of bacterial degradation. And the method for this was actually developed by Luke Wiley and a whole team at Imperial College London. So I think it would be a little much for me to explain exactly how it works. Instead, I'm putting up a paper here, and I'm hoping this conference will be a springboard for further discussion on it. But basically, we had about 3,000 plasma samples at 1, 3, 6, and 12 months of age, and which were run through LCMS, and through which we needed to do a whole lot of peak processing, and finally you get your metabolite concentrations. And so actually a lot of the time whenever you send your samples to people who do metabolomics, they can send you just the concentrations, which saves you a lot of headache, just so you know. So this is some work that I did with my master student, Paul Momo. And essentially, we just looked at, okay, is there any difference in these levels between children who are good growers and bad growers? So bad growers means they were experiencing stunting pretty consistently throughout the study, and good growers means they were above an HAZ of 2. And we didn't actually see too many differences in these levels, metabolite levels, between the two groups, except for one time point in kynurenic acid in month 3 and picolinic acid in month 3. There it was actually rather that higher levels were associated with poor growth. So this wasn't hugely informative for us, but this is not where you stop. You keep going. So we needed to know, for example, if the nutrition interventions had any effect on this whole pathway. And one thing we found is that, actually, yes. So in 6 and 12 months of age, which is key, the kynurenine and quinolinic acid levels were higher in children with the nutritional supplementation. So without any significant increase in the others. So it did seem like at least tryptophan was making its way down the right pathway towards the NAD+ synthesis. And on top of that, you had better growth. You had higher citrulline in children with nutritional supplementation, which is indicative of better gut environment. So this is consistent with what the trial was finding. What was interesting was that in the WASH interventions, we didn't see actually any positive effects really. In fact, except for, in quinolinic acid, epiclinic acid in month 12. So slightly higher levels of that in the WASH groups, but no effect on tryptophan or kynurenine or the KT ratio. And actually, oddly enough, higher inflammation and bacterial degradation in the WASH interventions. And actually, this is consistent with the study because, oddly enough, the study found that a lot of the children in the WASH group had higher pathogen prevalence. And that's a discussion for another time. So, we didn't see what we were really expecting with the KT ratio. So we needed to know, okay, was there any effect of parasite burden on this pathway? As you can see, the children in this study, they have actually a pretty high pathogen burden. These are just the top 5. And a lot of them are co-infected, already starting at 1 month old. So these are a type of beta coefficients from a particular kind of regression, a Tobit regression. I won't go too much into why we had to particularly do that, but at month 1 we didn't see any association with the metabolites. But at month 3, 6, and 12, we saw, quite a few things starting to spark up. And in the interest of time, I'm just going to fly a little bit through it. So of note, nothing from E. coli. These were all non-diarrheal samples, but considering the things that we've seen with respect to E. coli, non-diarrheal E. coli, and all the things they can cause, we were surprised that it basically had 0 effect on the tryptophan pathway. However, Cryptosporidium and Campylobacter appeared to be quite active at different times of the month. And particularly, we were interested to know this Cryptosporidium and Campylobacter increases in the inflammation marker neopterin, the kin-to-tryp ratio, which is also an increase in inflammation, but then actually also increase in the quinolinic acid to tryptophan ratio, which means that it is somehow making its way down to the key molecule to make NAD+. And then I remembered this actually is not too surprising because sorry. Yeah. Because actually one of the markers of Campylobacter in a previous study also done by John Swan was an increase in these sort of byproducts of nicotinamide degradation. So it seems to be sort of sequestering NAD plus byproducts of nicotinamide to itself. So to actually further get some insight into, now that we know that that the pathway is affected by these pathogens in a particular way, there is a further study underway in which another cohort, actually a cohort from a previous MALI-D cohort, will receive or has received treatments for bacteria and the particular protozoans that you saw in there, as well as a nicotinamide supplement, and to see if basically this can help supplement this pathway. So a bit similar to the previous study, we get one or the other or both. And what we're actually currently doing right now is we're engaging in it using both untargeted metabolomics from the urine and targeted metabolomics, where we'll again look at the tryptophan assay, but then actually also we're doing a kind of a much broader panel on the plasma that includes also the indoles that are involved in the inflammation pathway, quite a few lipids, and just a whole slew of molecules. So I know that was a little bit of a whirlwind. I just wanted you to see some of the things that are possible. And also I just wanted to reemphasize what Brian said, which was like it takes a whole team of people to generate these results. There are many groups involved. I really want to thank you, Brian and team, for organizing this. I think it's great. I've put down here the funders because you have to, but I've been acknowledging people who are involved throughout my presentation, because like I said, I really want you to see the faces behind it. And I just want to just give one last thank you to faces you don't normally see in the papers, which is basically the moms who have to trust us with their children. These moms are amazing. They are vigilant, but they're very much interested in the health of their children. And so the SHINE trial that I mentioned earlier on, it just came out with a paper. They had a very hard time getting very strong associations between their interventions, except for the nutritional intervention, and any measures of improved growth outcome. But they recently also analyzed, a mother's autonomy, education, etc., to see, if that had anything to do with growth outcomes, and those associations were huge. So, just a shout out to the people who keep the world running. Thank you very much. I'm very, very open to your questions. I understand I went a bit over time, but I have time, so I'm quite happy to hear your questions.
No worries, Gardana. Thank you very much for a very interesting lecture. It's really promising what could this analysis could do when we start to review the effectiveness of our interventions, right?
It's also good sorry, Francis, just to add, it's also good to know that we do have birth cohorts as well, being birth cohort studies being run by FNRI. So
that is right.
The examples that you presented, Gordana, I hope it inspires FNRI on what studies we can do. So these growth outcome studies on longitudinal studies. So just wanted to add that.
Yes.
Great.
Okay. So before we proceed, I would like to acknowledge the presence of our director, DOST-FNRI Director 4 and Scientist 2. Also our one of the pillars in nutrition and a scientist, our director, Dr. Imelda Agdepa. Maagham na hapon sa inyo po, Dr. Agdepa.
Good afternoon, everyone. Yes, good afternoon. Sorry, I just checked in late. It's just because of some other official matters. Thank you so much. Thank you, Gordiana, for that for that lecture. I was listening since then when you started. Thank you so much.
Thank you. And I know I didn't put in a lot of things in methodology, but as understood, this is a bit more to inspire what can be done. And I know that Brian's lecture and a few other lectures after me will go much deeper into methodology.
Okay, so we have a couple of questions for you, Gordana. Let's start. Okay, so first we have from Yas Junio of UP Diliman. Her question is, what is your basis for your preference of using SIMCA for your statistical analysis?
Okay, thank you. So I used SIMCA in the first study, and initially the basis was simply because it was easy to use. So that's when I started to collaborate with John, and he was teaching me metabolomics, and I was still doing my PhD, and it is one of those click and select tools. That being said, you need a license for it, so it is expensive. You don't need it. And actually, I have since stopped using it because I no longer need to use a click, like a quote unquote user-friendly click-based thing. That being said, though, I have to say that the methodology behind SIMCA, like the methods that the people who made SIMCA that they use are pretty sound a lot of the time.
All right. Next, we have a question from Jason Alcano from Central Mindanao University. So actually, he has 3 questions. So first, is it recommendable to report unknown but detected compounds in a metabolomics study? And then his second question is, what is the premise for reporting or not reporting these unknown compounds? Will it cause a difference if we omit them? Thank you very much.
Well, that's a good question. What I've seen in the literature is actually a bit inconsistent. It seems like people sometimes go on preference. I think it's best to report unknown compounds because it's pretty easy to do it. You just say unknown compound number 1, the feature is at peak shift 1.12. And you know, it goes to show that you've detected stuff that maybe you're, you know, we still don't have an easy way to identify it. And it's okay to not want to go further identifying it. If it's not your goal to identify every single thing that's in there, then, you know, and if it's pretty difficult, it's okay to not go there. I will say one thing. Recently, there's been like a panel that has come out with a little bit of, like, I guess, recommendations on what you are allowed to describe as annotation versus identification. And annotation is basically when you've seen a compound there and you use tools to identify it, but you haven't gone the full route to absolutely confirm its identity. And the full route is to spike your samples with a standard increasing dose, and then you are fully confirmed. But most of the time in these large untargeted studies, you don't really have the resources to spike every single thing. So all the things I identified are still considered in untargeted as annotated.
All right. Next question would be coming from Professor Marites Cation from UST. Have you looked at which phase of the disease is Preziquantal best administered to patients after identifying these metabolites?
Thank you. That's a great question. So actually, it's already pretty well known that Preziquantal does not work very well at earlier phases of the disease. So it's okay when you first get infected, but as it's traveling through, it's actually not very effective, especially at the lung stage. So all the people that were enrolled in the study had a patent infection. That means they had a full adult infection. And the way that we knew that was that they were passing eggs in their stool or urine. So yeah, and we wouldn't have done the study with other stages because we know already that Preziquantal is not very effective at those stages. I will add that, of course, obviously there are some groups. So Jennifer Kaiser, who was my previous supervisor, she is one of a handful of people who are trying to develop new and better medications. But that is a long road, and that's what I was doing before I moved on to metabolomics.
Another question would be coming from our colleague, Mark Pretzel Sumaraga, who is now in France for his PhD studies. He's asking, may I know if there is a way or a consensus to select a cutoff for which metabolites are significantly associated with the outcome?
Yeah, that's a great question. So I wish actually I actually do have a, we ran a workshop in Fortaleza a year ago where I went over that, and I'm wondering if I can, you know what, it's going to take a minute to pull up that slide, but basically, just going to pull up one figure. Okay, so just I know this is a little bit of a complicated figure, but basically all the features here pointing upwards are associated with higher levels of Preziquantal in the plasma, and all the features pointing downward are associated with lower levels. Now you see here, there's, first of all, for me to even be showing this model, I would have to be sure that the model has significance. So this is the loadings plot of an orthogonal partial least squares analysis, which is basically like, it's like a PCA, but where you regress the PCA against your variable of interest. In my case, it was how my variable interest was the level of Preziquantal in plasma. And for us to be sure that that model is significant, we have to basically rerun it with like a sampling and resampling of the samples, a kind of a cross-validation. And that gives you like a predictive capacity score, Q squared of the model, which doesn't really have a cutoff, but it should be positive at least. And after that, we basically have to run a kind of another bootstrapping model to ensure that the model itself is significant. And that bootstrapping basically requires like mismatching some of the values and seeing if the things with the correct values has the same significance as the thing with the incorrect values, if that makes sense. But then on top of that, you can see here that my spectrum has some things that are colorful and some things that are black, and all things that are black are not significant. And that cutoff is basically, Brian had alluded to this earlier. There's a few different corrections you can use to just make sure you're not getting any false positives. And one way is to sort of divide the p-value by the number of features measured. So that's kind of what's used here. But there are a few other methods as well. I know that's a very long answer, but that just kind of goes to show you that, yeah, you have to learn a few steps.
All right, a clarification from Dr. Sofia Amara. You mentioned earlier that washed children had higher levels of pathogens than non-wash. Is this correct?
Yeah, so not all, I should clarify that, not all pathogens. So it was a very unusual finding, like it was highly unsatisfactory to everybody involved. But yeah, in this particular stages, the wash intervention did not at all help with viral and bacterial infections. And in some cases, those were higher in wash, but it did actually help with parasitic infections. So like protozoans, Cryptosporidium, etc.
All right, okay. So for those questions that we have not answered online, we'll try to still secure your questions. So you can still post your questions here at the chat box, and we'll accommodate them, and then we'll forward them to Gordana so she can answer them, and we will be contacting you for the responses. Okay, so
thank you.
Why not let's give a virtual clap to Gordana for that wonderful presentation. Can I see a clap reaction from everyone? Come on.
Thank you, Gordana. Thank you.
All right.
I mean, her work also shows that it's really an army. You need a lot of people if you want to pull this off. So I'm happy that we have a lot of participants today, and hopefully this starts a collaboration amongst all of you, because it's not easy.
All right. Okay. We'll have a 5 minutes break before we proceed to our next topic. So stay tuned and see you in a bit.
Thank you, Gordana.
In 2010, DOST-FNRI revolutionized its take on nutrition by incorporating genomics in its researches, investigating the relationship of anthropometric, biochemical, clinical, dietary, and health components to the genetic makeup of Filipinos. DOST-FNRI is looking into providing a more personalized approach of achieving optimum nutrition to every Filipino. Through nutritional genomics, we attempt to better understand how a single nucleotide, a single letter, can make a big difference in our way of living. And so, to support this endeavor, DOST-FNRI capitalized on establishing its molecular laboratory known as the Nutritional Genomics or the NewGen Lab. With its thrust of bringing science for the people, the NewGen Lab provides assessment of genes and genetic variants associated with micronutrient deficiencies and diet-related non-communicable diseases. The NewGen Lab is equipped with some of the state-of-the-art equipment in molecular biology, such as the automated nucleic acid extraction machine, bioinformatics, real-time and digital PCR systems, automated liquid handling system, micro-volume spectrophotometer system, and gel documentation system. With a complete workflow adhering to the highest standard of competency in laboratory testing, NewGen Lab is setting the mark in providing mole biotechniques with transparency, validity, and reliability. Whether you are an individual or a group of enthusiasts seeking answers in nutritional genomics, the NewGen Lab awaits you. Our proficient personnel works in close contact with our customers through customized analysis, provision of technical consultancy in nutritional genomics, and standardization or laboratory competency. To even serve you better, we are partnering with a leader in genomic testing for a higher throughput and faster turnaround time. Welcome to the new generation of nutrition. Welcome to the NewGen Lab.
And we're back. Now, our next topic will be given by none other than our DOST Balik Scientist grantee, Dr. Gerard Brian Gonzalez. For those of you who just joined us today, Dr. Gonzalez is currently an assistant professor at Wageningen University and Research, the Netherlands. Our speaker has been in collaboration with various institutes, organizations, and hospitals, such as the Institute for Metabolic Science in the University of Cambridge, the Hospital for Sick Children or Sick Kids in Toronto, Canada, Kenya Medical Research Institute, and the World Health Organization European Regional Office under the Nutrition, Physical Activity, and Obesity Unit in Copenhagen, Denmark. Dr. Gonzalez specialized in metabolomics and food science and technology. As a food scientist, he uses his skills and expertise in food and nutritional chemistry, specifically mass spectrometry-based analysis, to study severe acute malnutrition in children from a molecular perspective. I now turn over the virtual floor to Dr. Gonzalez, who will be talking about mass spectrometry-based analysis of the metabolome. Over to you, Dr. Gonzalez.
Thank you. Magandang hapon ho. Magandang hapon sa inyong lahat. And thank you for the kind introduction, Francis. I don't know where you got that. I actually didn't give you a profile. So thank you. That's very kind of you. All right, so for today, just a disclaimer. I know we have 247 participants, but my next lecture is gonna be quite technical in a way. So this will be a presentation about mass spectrometry. And if any of you think that this is too advanced, so probably people who do not have any chemistry background might find this lecture too advanced. And yeah, you feel free to jump out of the meeting. It's fine. And for those people who already are working with mass spectrometry, this might be too basic. So feel free to also jump out. But if you are interested anyway with mass spectrometry-based analysis, then yeah, stay tuned. So these are, yeah, I will be speaking slower than I usually do just to make sure that this become understandable. So I have alluded to this slide on Monday, metabolomics instrumentation, and in Gordana presented these two examples very well. We usually use two types of instrumentation when we do metabolomics. I said usually because, of course, there are many other techniques that you can use. Not many, but there are other techniques you can use. The most popular one, I would say, is mass spectrometry. It has the advantage of being more sensitive, and it's good for metabolite detection of small with lower concentrations. But in terms of quantification, it's very good. But when you deal with very large scale samples, then you will have a lot of problems with stability of your analysis. Whereas when you use nuclear magnetic resonance, as what Gordana gave you as an example, the quantification is very good. Structural elucidation is also very good. But yeah, it is less sensitive. So when you use NMR, you tend to prioritize those metabolites that are of higher concentrations. Whereas if you want to look at a lot of molecules that are really smaller in quantity, then we usually go for mass spectrometry. By the way, the apartment next door is doing some renovation, so you might hear some banging. It's not me, it's the neighbor. So sorry about that.
Copy that, Brian.
Well, they didn't ask me permission. So all right. Yes, so today's lecture is about basic principles of mass spectrometry. Yeah, the very dictionary definition of mass spectrometry is that it measures the masses of individual molecules that are converted to ions and But simply speaking, mass spectrometry, if we want to understand it, we just need to break it down into its individual parts, and that's mass, spectro, and metry. I have to emphasize that it's not mass spectroscopy, and it's very annoying for people to call it mass spectroscopy, because when you say spectroscopy, it's usually an optical technique. So things that use light. In mass spectrometry, we don't use that. So mass, as we know, is a mass. Every matter has a certain mass. Spectro is a range, and metry is the measurement. So mass spectrometry, very simply put, is the measurement of a range of masses. Now we have to understand. Do you see my cursor, by the way? Do you see my cursor?
Yes, Brian.
All right.
We're seeing your cursor.
So you have to understand that when we say mass in mass spectrometry, we don't really mean the actual mass of the compound. What we mean is the mass to charge ratio, and we will get to that in a bit. But mass spectrometry is really just the measurement of a range of masses. How do we do about with mass spectrometry-based metabolomics analysis? So this is the basic skeleton of a mass spec. So you have a source. We will go to all of these individually later on. Then it gets, you have your analyzer, then you have your ion detector, and you have your computer over here. And what you get out of a mass spec is an accurate mass, again, based on a mass to charge ratio. I will explain that in a bit. But normally we don't, you don't inject blood in a mass spectrometer. You can directly infuse your sample, but you first need to isolate or extract your metabolites. So we normally do that if you put, let's say, methanol. You can do a Fulch extraction, for example, where you do chloroform, methanol, water. So you have to remove the metabolites from the sample, and then you can either directly infuse it to the mass spec, which some people do, but most often we use a chromatography. We attach our mass spectrometer to a chromatographer. So either a gas phase chromatography or gas chromatography, or a liquid chromatography. And the point of this is to separate your compounds so that they don't go all together in the mass spectrometer, and it just helps us more understand what's in our sample. So questions we get when people want to do mass spectrometry: can we measure anything with a mass spec? Well, the answer is no. We can only measure, based on the definition, only those that you can ionize. So it's a method of ionizing your compound. So if you have a compound that cannot be ionized, then you cannot detect it with a mass spectrometer. Another question is, can we quantify anything with a mass spec? Quantification requires an additional step. Gordana was mentioning this a while ago. You really need to know what you are measuring. So the validation of your metabolite is very important, but also you need to have a standard for your compound. So only those with suitable standards that are available, you can absolutely quantify. Well, you can semi-quantify some of your standards without some of your metabolites without the suitable standards, but you cannot call it an absolute quantification. So by quantification here, I mean, you really know the micrograms per ml of urine or micrograms per ml of blood, for example. You can only do that if you have a suitable standard. Otherwise, it becomes a pseudo quantification or an untargeted or a semi-quantification kind of study. So as I said, normally we attach a mass spectrometer to a chromatography technique, and that is because we want to make sure that not all the compounds go to a mass spectrometer. It helps us with identification. It helps us with a lot of many things, such as matrix effect, such as ion suppression, for example. So as I said, when you do mass spectrometry, you need to ionize your samples, but sometimes some metabolites reduce the ionization of other metabolites. So it would be interesting to separate them first before introducing them to the mass spec, so that we optimize the ionization of your compound. So chromatography is simply the technique for separating different components in your sample. We know very well of this paper experiment where you put ink and you immerse the ink in some sort of solvent, like an alcohol, for example, and then as the alcohol goes up, the colors of the ink separate, and they turn into their individual color components. So that's basically what you have with chromatography. And how that works is that we exploit the differential interaction of the analyte, in this case, the colors, with either the stationary phase or the mobile phase. So in both liquid chromatography and gas chromatography, you have both a stationary phase, which is the column. So this is the column here, the column. And you have a mobile phase. In the case of liquid chromatography, it's your solvent, so either methanol or water. When your samples are injected to the system, your liquid mobile phase pushes your sample through the column. And then because of the interaction of your compound to the stationary phase, which is the column, and also the solvent, then your sample, your metabolites get separated. It's the same principle as in gas chromatography. The only difference is that in gas chromatography, your mobile phase is gas. So when you have a gas mobile phase, that's a gas chromatography. Liquid phase, you have a liquid chromatography. Easy breezy banana. So when you deal with liquid chromatography, for example, we have to understand even the most untargeted analysis, there's no such thing as a real global metabolomics for one sample, because metabolomics, even if it's untargeted, it's very platform specific. So you cannot analyze all the metabolites in one run. There will be metabolites that are very polar, for example, things like glucose and some organic acids. They will not retain very well if your stationary phase is non-polar, for example. And you can also have very non-polar lipids, for example, like triglycerides, and those are rather difficult to separate if your stationary phase is polar. So we use different column chemistries depending on what type of metabolite you would like to measure. So before, when you want to measure really hydrophilic compounds, we use a normal phase column. When we want to target, let's say, lipids or non-polar compounds, we use a reverse phase, which is just the reverse of the normal phase. And very recently, there's a new column chemistry. It's called hydrophilic interaction liquid chromatography, HILIC column. It is very similar to a normal phase. You have a polar stationary phase and a non-polar mobile phase. But the difference is that in normal phase, we usually use more stronger solvents, like really pure organic solvents. Whereas with hydrophilic interaction columns, we can use less non-polar solvents that are miscible in water. But that depends. So for example, if you want to separate things like sugars, then you would normally use something with a stationary phase that is more polar, so that your compounds are stuck there. So if you want to measure, let's say, TCA cycle metabolites, you would have a better chance of analyzing them if you have a polar stationary phase. If you want to study lipids or bile acids or flavonoids or these other secondary metabolites, you'd better off having a reverse phase analysis. So how do we ionize compounds? As I said, when you want to use mass spectrometry, we need to have the ions. We need to have the compounds ionized. So after they are separated by the gas chromatographer, they need to be ionized in the ionization cell. And in gas chromatography, there are two types of ionization techniques: the electron impact or electron ionization, or the chemical ionization. By the way, I have to say that for gas chromatography, you use it when your compounds are volatile. So when they turn to gas. But if they are not volatiles, then you can make them volatile by derivatizing them. So for example, when we measure fatty acids, they are not volatile. So we can make them into fatty acid methyl esters, and then they become volatile. Then you can separate them in a GC. And a GC analysis is very good for smaller compounds. They tend to separate better. And the chromatographic resolution of a gas chrom is superior, far superior than a liquid chromatographer. So how does this work when you want to use a GC mass spec? In an electron impact, the mass spec bombards your analytes after being separated from the gas chromatography with electrons, and that impact charges your compound. So you now have a charged compound, charged analytes that will go to the mass analyzer, so to the mass spec. Chemical ionization, on the other hand, is a softer way of ionization, where instead of the electrons bombarding your compound, it bombards a reactant gas, and that causes a chemical reaction that gives a charge to your compound. So now you will have charged metabolites that can then go to the mass spec. So it's rather straightforward because gas chromatography is already in the gas phase, and mass specs only analyze things in the gas phase. So only in the gas phase, mass spec. So you might be wondering, how does it then that it works on a liquid chromatography? Well, if compounds can only be analyzed in the gas phase, liquid chromatography first works by converting your liquid into a gas phase. And one of the most common, or if not the most common way of doing that in mass spectrometry-based analysis, metabolomics, is with the use of electrospray ionization, or ESI. So suppose this is the LC. Yeah, sorry, my penmanship is not very good when I'm writing this way. So suppose you have an LC, and this is the flow of your liquid. So the compounds are being separated already, and it's introduced now into the mass spec. So what happens is that the liquid now gets in contact with two plates, or like this is actually a round plate, but with a plate that has a charge. And this charge, depending whether you can charge it positively or negatively charged, the charge transfers into a droplet. And this droplet, because this is enclosed in a heated chamber, this droplet becomes smaller and smaller. And when that droplet becomes small enough, it reaches what you call a Rayleigh limit, whereby you have a very high charge density. So positive charges are very much concentrated in a small droplet. So what happens, as you know, if you have the same charges that are very close to each other, it's like a magnet. What happens is that they repel, they move away from each other. And that's where you have a Coulomb explosion or Coulomb fission. And that fission transfers the charge to your compound. So now you have a charged metabolite, and it's now ready to go to the mass spec, right? Because this can get very technical. So I welcome people to ask questions, and I'll try to answer them as we go. So if you have questions so far, go for it. And I hope you're still there. Yeah, okay. It's very difficult when you don't hear anything. Yes, so that's your liquid chromatography mass spec, how to convert liquid, and then it becomes a gas phase using the electrospray ionization to get ionized metabolites, and then your metabolites gets analyzed in the mass spec. There are many kinds of mass analyzers. So you have low resolution mass specs and you have high resolution mass specs, and that depends on what you need to do with your metabolomics analysis. So the most common low resolution one is a quadrupole. If you have three of those things, then that's what we call a triple quadrupole. And probably I'm sure that many of the labs there in the Philippines have a triple quad. They are, of course, cheaper because they have a low resolution. But if you have a lower resolution, not slightly, but cheaper machine, are you not able to do metabolomics? No, you still are able to do metabolomics. And that's what Gordana was mentioning a while ago. You can do targeted metabolomics. And when we do targeted metabolomics, it's usually done with a low resolution machine because they're very sensitive and they're very good for quantification. So if you want to measure very low abundant metabolites, vitamins that are very low in abundance, that's when we usually go for a triple quad analysis. But the difficulty with a low resolution machine is that it's not good for identifying unknown compounds. And I will get to that in a bit. There is where the high resolution machines become very helpful. There are two very common types of high resolution mass specs in metabolomics. We call them time of flight or an Orbitrap. By the way, I'm not sure, is anybody from the PGC here, PGC Diliman, from the PPMF? Can you make yourself known in the chat? And I know Hias is here, so that's good. Anyone from the LSU, the metabolomics facility, is Karen here? Yeah, so later on, I invite you to join the Q&A, Hias and Karen and people from PGC, so we can all have this together. So that's great. So we have two kinds of machine, and usually there is another one, FTICR, for example, which is also a high resolution machine. And I'm happy to say that we have both of these types of machines now in the Philippines. So we can, in the next 8 days, get inspired by the examples of our other speakers, and we can try to replicate them with our own samples in the Philippines. So when you have a high resolution machine, it's very good for untargeted analysis. In fact, it's what we use for untargeted analysis. And you have a higher confidence of identification when you have a higher resolution machine. That being said, the sensitivity could be, many high resolution machines are already very sensitive, but still the sensitivity of a triple quad is still very superior to a high resolution machine. So this is a scheme that I presented last Monday. As I mentioned, if you want to go targeted metabolomics, the stability and repeatability of your sample becomes very high, but the amount of information you get from it tends to be minimal. Whereas when you do untargeted metabolomics, you get a lot of information, But as Gordana mentioned a while ago, you need to use a lot of bioinformatics and multivariate statistics to get sense of the data. But with regards to mass spec use, usually the more untargeted we go, the more high resolution we use, and the more targeted we go, the more low resolution we use. So there's a question here of what is the usual gold standard for mass analyzers for quantification. So as I mentioned, as I mentioned a while ago, for quantification, if we only want to quantify compounds that we already know what they are, you can do that perfectly with a low resolution, triple quad machine. I'm not saying you cannot quantify with a high resolution machine. You can, and you can do it very well. It's just that the sensitivity is far superior in a low resolution machine compared to a high resolution machine. There's no gold standard. There's always a fight between, whether we use a time of flight or whether we use an Orbitrap. Of course, Orbitraps have a higher resolution, but there's always a pros and cons with that. We have another question. I mentioned a while ago that the suitable standard, I would like to ask what do you specifically mean? Could you cite some example? Well, a suitable standard is the actual compound. So if you want to measure things like tryptophan, you need to have pure tryptophan standards in your lab. Otherwise, you won't be able to really absolutely quantify tryptophan. You cannot, for example, use a standard of tryptophan to measure something else like proline. You can, but it's really best that you have the actual standard for your sample. What we also sometimes do is that, you cannot buy all chemicals. For example, if you want to analyze polyphenols with different glycosylation patterns, we don't have standards for all those. So we do what we call a relative quantification. So relative to a suitable standard, we can quantify more or less the other compounds. But so that's what I mean. So when I say resolution, what do I exactly mean? So resolution is the ability of the machine to distinguish two different masses. So this is a very good example. When you have a low resolution, we have one peak here. This is the mass to charge ratio. I will get to that in a minute, but when you have your machine here, it's 287.1823, for example. In a low resolution machine, we can notice that as you increase your resolution, in a low resolution, we see that there's only one compound here or one feature, but actually the more high resolution we go, then we see that, in fact, this one peak here is composed of two peaks. So you can see here, this is the higher resolution. You can really separate compounds at the 2 decimal points in the mass. Whereas if you have a low resolution machine, then it's rather big, and you will, if you have compounds that are very similar in mass, like these two, then you won't be able to distinguish them in a low resolution machine. That being said, we use high resolution machine because it has more accurate in terms of mass. And you're really able to distinguish metabolites. So for example, if you use a low resolution machine for your metabolomics, you might think that this is a glycylysine because of the mass. But in fact, when you use a high resolution machine, you will find that this peak here is not correct. In fact, these are two different compounds and they have two different identities, right? So the higher the resolution of the machine, the more accurate the mass it gives you. And the mass is what we get from a mass spectrometer. I'll get to that in a bit. It will get clearer towards the end, well, I hope. Why is mass important? First, before we go into mass spec, there are two different definitions of mass we have to understand. The first is called average mass, or it's the sum of all masses. Basically, this is the mass that you get when you read a periodic table. So this is the mass where we take into account all the isotopes. So we know that carbon has a normally it has a carbon 12, but it also has an isotope, which is carbon 13, which is present around, I think, 2% in nature. So when you take that all isotopes into account, you get the average mass. What the mass spec measures is what we call the monoisotopic mass. So it gives you the masses for particular, most especially the most abundant isotope in the of the molecule. So let's give this example. For example, quercetin. This is a polyphenol. This is a flavonoid found in onions. Found in it. It's a very healthy flavonoid, anti-cancer, anti-inflammatory, anti-terrorism, anti-everything. So it's very good. The average mass of this molecule, if you just look at the periodic table, is 302.236. But if we only consider the most abundant isotope, then the monoisotopic mass is 302.0426. Again, we have to understand that this is a mass to charge ratio, and that means that the mass of the compound is divided by the charge that it gets. If you remember in the electrospray ionization a while ago, we charge the compounds. Luckily, in metabolites and metabolomics, they only carry one charge. So the mass to charge ratio is mostly the charge itself, but we have to understand it is charged. So instead of this is the actual mass of this compound, what we see in the mass spec is not 302, but 302 plus the charge or minus the charge. So plus the charge if it's positively charged, because we add a proton, and it's so it becomes 303 in a positive charge. It becomes 301 in a negative charge. And why is this mass very important? So the more accurate this mass, the better we can identify an unknown compound. So let me give you an example. If you have, let's say, quercetin, and for example, you analyze it in a low resolution machine where you have a lower mass accuracy. For example, you see 302.05. You will see that we have a lot of hits possible. So by the way, here in this case, we can calculate the mass based on the composition of their atoms. But if you also have the mass, we can then, mathematically infer what is the elemental composition of that compound. So if you have a mass right here, 302.02426, we can make some calculations. We can make combinations of carbon, hydrogen, and oxygen, and we can come up with a putative, how do you call that? Elemental composition or this elemental structure, not structure, but elemental composition, a formula. I mean, the elemental formula, the molecular formula. So once you have a very high resolution mass spec and you get the mass, you can use that mass to try to figure out what is this compound. So there are softwares online with the MF Finder, for example. If you put the mass that you found in your mass spec, again, you either have to add your proton or remove your proton because it's charged. You can see that if we have a 24 ppm error in your mass, then you have a lot of hits, possible different elemental compositions. But the higher, if you have a much higher mass accuracy, as you can see here, we add more decimal points to our mass. Then we now see number 2 right here. This is the mass. This is the compound that we were looking for. Yeah, that's the compound. That's quercetin we're looking for. So the higher the resolution, the higher the mass accuracy of your mass spec, the more you are able to find out what this compound is, this unknown compound is. But as you can see here, this is already very high resolution, 0.1 ppm error, 4 decimal points, and you still have 2 compounds that are, closely resembling the real mass. So you have either if this is the mass that you found in a mass spec, then the identity of your compound can either be a C14H4N7O2 or a C15H10O7. We know that it is C15H10O7, but if you're analyzing unknown compounds, then you probably don't really know. You don't probably know what really is the compound. There's a question here: is it possible to have multiple charges on metabolites? And thank you very much. Yes, it's very correct. For small molecules, it's usually just one charge. For proteins, you can have a lot of different charges. And luckily, we're not analyzing proteins now, so only metabolites. So how do you know which one is the real compound then? If you have a already very high resolution, well, there are machines out there that are even higher resolution, like 1 million resolving power. Or so, for example, here you already have 4 decimal points, and you have still 2 candidates. You can go all the way to, let's say, 6 or 7 decimal points, and then you will have your here your actual compound of interest. Yes, thank you for letting us hear that. So
Sorry
yes,
about that.
no problem. So yes, you can go as high resolution as you want, or you can then use, the liquid chromatography. So hopefully these compounds separate in different times in a chromatography. You can then buy in the standards, the suitable standards, and you can inject that on your sample, and then you will be able to verify what the real identity of that compound is. Or what you can also do is look at the fragmentation of the masses. So those ions that are ionized in the mass spec, you can trap them and bombard them with a very high energy, such as like an argon gas, for example, and that molecule will start to break into smaller pieces. So this is quercetin again, the same compound. As you can see here, it's 303. What we were analyzing a while ago is 302, right? But what you see in a mass spec is 303. And again, that is because you are charged. So you add a proton to your mass. And that's why what you see is 303 in a mass spec, but in real life, it has a 302 mass. We can then identify the compound or annotate the compound. As Gordana mentioned a while ago, we can annotate the compound, meaning we're not really sure, very, very confident about its identity, but we can compare the fragmentation of this with other compounds. So there are databases available where they collected what is the fragmentation pattern. So these patterns here are characteristic of quercetin, for example. So if you have an unknown compound and you analyze it with a mass spec and you don't know what it is, you can then do spectral matching, for example, on databases. Next week, Monday, we will have a very good lecture, well, hopefully, with an expert on metabolite annotation. So I will not even pretend that I'm an expert on this. We have Justin for that. He will teach us about GNPS and other spectral matching techniques on how to identify unknown compounds. So there's a question, will different configurations of the same compound elute differently in the LCMS? Well, hopefully, more or less, yes. So if you use different columns, for example, if you use different pHs, you can play around with the temperature, the flow rate. You can do a lot of different things with a liquid chromatography, and then hopefully it separates. If it doesn't, then you need to find other ways. Yas is saying here in the chat that chirality could be an issue, and it is true, but we also have chiral columns that can separate them. But chirality is, of course, difficult. I have to stress out that in a mass spectrometer, we get is a mass. And there are compounds that we call isobars or isomers, meaning that they have exactly the same mass. And the most popular example of that are 2 amino acids, leucine and isoleucine. They have exactly the same mass. So if you don't separate them in a liquid chromatography first, you will not be able to identify which one is isoleucine and which one is leucine. And that's why it's very important for us to have a liquid chromatography before we do the mass spec. And of course, a lot of flavonoids have the same mass to charge ratio, so M over Z value. That's very good, especially if you have a flavonoid where the sugar is attached in different aspects. So you can have in different sites. So you can have a sugar that's attached here. You can have a sugar attached here. You can have a sugar attached here. The mass is exactly the same, but their fragmentation pattern will be different. So that's how you are able to identify them. All right. So yes, there's a comment about it. Yes, the fragmentation pattern will be different, and that's how you are able to identify them. Again, Monday, stay tuned. We have a lecture about spectral matching and how to use GNPS from an expert, one of the developers of this platform. So it's very good. How do you know what adduct is in effect for the metabolite? Is it done manual annotation or software identifies it automatically? Well, adducts are the ones that are attached to the metabolite. So this here, H, for example, that's called an adduct, an H adduct. But other adducts also attach themselves to the compound. So you can have a sodium adduct, or you can have ammonium adduct, or so many potassium adducts, so many different things. Well, you can identify that based on, with the fragmentation. Sometimes the adducts get released. And sometimes what we do, there's a software that I use, for example, Progenesis. There's a freeware called Camera. For the same compound, we can then know whether this compound is the same based on different adducts, because the mass defect is different, or the mass difference is. So when you add a sodium, you know that there is a particular mass of sodium that's added to your compound. And if you remove that mass and it matches with another compound where you removed the other adduct, then that's how you know that this is not the real mass, but the mass of an adduct. Hopefully, I think tomorrow we can, if the software is running well, then I can show it to you. But yes, this is just a very brief intro of how mass spectrometry works, in fact. If you want to do mass spectrometry-based analysis, it's not an encouragement, but I demand, I asked you that you should talk to a chemist, and we have experts in the Philippines who you can have a discussion with on this. You should never do this alone. Metabolomics, you should never do alone anyway. So this is the workflow of a mass spectrometry-based analysis. So when you have a blood sample, for example, you isolate or you extract your metabolites from the blood matrix or the urine. You separate them using the LC. It gets electrosprayed, so the liquid turns into a gas phase. And then it enters the mass spectrometer, for example, where the masses are measured. So what you get from a mass spectrometer is this. This is called a chromatogram. So each spike here corresponds to at least one M/Z value. And what we do, in fact, this is sample 1, sample 2, sample 3, for example, using some sort of bioinformatics, which we will discuss hopefully tomorrow. We can convert this into a workable table. So in this table here, we have, let's say, each row is your sample and each column is every M/Z value that is detected in your sample for every retention time. So when you do untargeted metabolomics, you can go to up to 2,000 different variables, 3,000, even more, thousands of variables per sample. And to make sense of that, then you need to use what Gordana was saying a while ago, statistics, informatics, and machine learning to make sense of this. As Lars mentioned yesterday, the biggest bottleneck now for metabolomics is identifying your metabolite. There are many tools. As I said, on Monday, we will have a lecture about that. But the tools that I mentioned a while ago are the simplest ones. So, when you have exact mass, you can then look at you can calculate what is the elemental composition. You can then use online tools to compare your fragmentation spectra. And then hopefully, after you have analyzed this table, you can then extract your biological information using some pathway analysis and stuff. So that is how we do liquid chromatography-based mass spectrometry. On Thursday, we will have a lecture, I think it's Thursday, with John. He will be showing us how they do it using an NMR. And on Friday, we will have a step-by-step metabolomics exercise with Dr. Laura Righetti. So stay tuned on that. But hopefully this gave you a brief introduction of how we run mass spectrometry-based metabolomics and how a mass spec works. All right, so I'll stop there to give chance for some questions and some interaction with the experts. Again, I have to say that we have experts in the Philippines for this. So yeah, we can then contact each other and ask each other questions later on. So I will stop there and I'll give the floor back to our moderator if there are unanswered questions.
All right, thank you, Dr. Gonzalez. Yes, I was I got hooked to your presentation because I am working inside the laboratory. And of course, when I see machines and instruments, that really got me hooked.
Yeah.
It's a gruesome process, if I may say, but the information, especially on your last slide, no, the information that we can have is really promising, especially using the tools of informatics. No, so I can't wait to have some of the projects here in FNRI to be rolling and incorporating metabolomics, and to do further collaborations with some of the experts in metabolomics here in the Philippines and our extinguished chemists here in the Philippines. Okay,
Yeah,
so let
you
me
have
just...
a lot, by the way. You have a lot. So
yeah. Okay. So let me just see some questions here. We have one from Christine Ramos from Biotech UP Los Baños. Will different configurations of the same compound elute differently in LCMS?
Yeah, I think I answered this question
Okay,
already.
you answered
So
already.
yes, the answer is yes, depends. You need to play around. Yeah.
Okay. Next, I think we have...
Yeah, I think the one that's here is yeah. Yeah, he asked,
Okay,
why don't you unmute yourself and you ask
Yeah,
the
yeah.
question and we can both have a discussion.
Yeah.
He asked, by
You
the
can
way,
actually-
is an expert chemist in mass spectrometry. So if you have questions,
Hi, yes.
Hello, good
Yeah.
afternoon. So Brian, I have a question.
introduce yourself first.
Hello, good afternoon. I'm Yas Julio. I'm actually the mass spec manager at the Institute of Chemistry in UP. So we have a QTOF, and hopefully by next year we will be working with FNRI and of course with Brian on some clinical samples. So I'm keeping my fingers crossed on that. So I have a question for you, Brian, because I just wanted to be what do you call this, up to date in terms of the terms. Is feature an accepted term now when you're referring to chromatographic peaks?
Yeah, so actually, yeah, as Gordana was mentioning a while ago, there is a consortium now trying to really put standards on how you identify metabolites.
Right.
The thing is, when we inject samples in a mass spec, you get tons of data, like 3,000 different peaks, right? And it's possible that only less than half, even only 10% of those are real metabolites.
Mhm.
Only 20% are real metabolites. So we cannot call them metabolites because they're not real metabolites. And that's why we call them features.
Okay.
Yeah, I think the name feature came from the data mining world, because we always use data mining anyway when we analyze, and they call it features. And that's why I'm not sure, but I think that's how it is. So to be safe, to not call them metabolites, which maybe they are not, we call them features or peaks. In fact, peaks or features. Feature sounds fancier.
Because some people, even some publications, they're very sensitive in the use of the term, especially annotation and identification.
Absolutely.
I think Gordana was making a point about that you have to differentiate between when you annotate, what does it mean to annotate versus what it means to identify. So
yes,
right. So some people are really very particular, but for people who have less experience in mass spec, that doesn't seem to have any difference, no? So
yeah, sure, yeah, sure, yeah. But yeah, I mean, in keeping with these consortium standards that I mean, these are conventions, of course. It's not like etched in stone. These are conventions. It's better to be acceptable with the I mean, it doesn't harm us to call it a feature. So
Yeah.
let's all use the same terms rather than having different terms.
Right. Thanks for that. Something new in the vocabulary.
Is anyone from, is Karen here, Lacerna? Maybe not. Okay.
Yeah.
Maybe not.
She was with us yesterday.
All right.
Okay, I think we have one question from Wilson Sumpio. I often see LCMS being used for vitamin D determination. How is it different from HPLCMS?
Well, the LC is liquid chromatography. HP is just, it is the same. It is the same. Well, of course, there is a slight difference when you talk about HPLC, which is the high performance liquid chromatography or high pressure liquid chromatography, with the ones we usually use more now, which is UPLC, which means ultra high pressure. So it's just the difference is just the amount of pressure that these machines can hold. So HPLCs usually have bigger columns. They require more solvents. When you use UPLC, then the columns are a bit narrower and smaller. The pressures are very high, and that's why we require less solvents. But they're basically the same.
Thank you. So do we have more questions? Any more questions from the group for Dr. Gonzalez?
I'm seeing none on YouTube, so I think we're okay.
Okay, so let's give a virtual applause to Dr. Gonzalez. Come on.
Thank you very much.
Thank you very much.
I'll just announce for tomorrow. Tomorrow we have another very special treat. My supervisor, in fact, and friend, Dr. Albert Coleman from the University of Cambridge will be with us. And he is the head of the core facility in the University of Cambridge Lipidomics and Metabolomics lab. He will be giving us his insights and giving some examples of what we do. In the afternoon, I have to have a disclaimer. So we use free online tools. However, it's possible that tomorrow the website is down. So in that case, depends on whether the system, the system online is available or not, then we may proceed. If not, I'll think of something else. But I think what I'm getting from this webinar, especially from Gordana's topic, is it would be nice to have an intro of what is PLSDA or what is PCA and all these multivariate tools that we are using. So if the tools are not available, then I will just give you instead an introduction on the multivariate statistical tools that we use for metabolomics.
That is duly noted. Okay, can we have some words from our director and scientist to Dr. Imelda Angeles Sectepa? Doc Mel?
Hello, Tata. Sorry.
Yes po.
Because I'm attending to a phone call. Sorry.
Oh, okay.
Ano
Apologies
sinabi
po.
mo?
If you have some words po after these two lectures.
Yeah. So I'd just like to say that hang on until the last day of this OMICS seminar. It's just because we still have a lot of special topics now for this webinar series. Wherein for us here in the Philippines, it's somewhat, still emerging, no? Except that except in maybe in UP, wherein we have the Genome Center. And also here at FNRI, we also have the Nutrigenomics Lab, but then we really need more knowledge and maybe the sharing of experiences to capacitate more our researchers and scientists here at FNRI. So join us until the end of the seminar or the webinar because we have more important topics. Maraming salamat and thank you so much for Gordana and also Brian, and of course to all the participants who are still with us until this It's already 5:10 here in the Philippines. So, so thank you for your time. Over to you, Tata.
Okay po. Thank you very much, Dr. Depa. So this, Brian, do you still have other announcements? None on your part. Okay. So on behalf
Thank you,
of
Tata,
US,
and the group. Thank
yes
you
po.
for
Thank
putting
you,
this
Doc
up,
Nel.
no?
Yes po. So on behalf of the DOST-FNRI technical team for OMICSena, this is Frances, and I would just like to say that stay safe always. Bye for now, and hope to see you tomorrow for another fun-filled lectures on OMICSena. So thank you very much, and please answer or accomplish your evaluation forms. This has been sent to your email, I think. So you can get your certificate of participations, and your certificates will be available starting October 20. Okay, so that's it again. Maraming salamat, maagham na hapon again to all of us. Stay safe, everyone. Bye.
Thank you. See you tomorrow.
See you tomorrow.
Doon.